DNA shuffling is an extremely powerful approach to artificial evolution of the genes and thus the proteins. This technique generates a library of mutant genes that will encode proteins with improved or unique functionality. DNA shuffling is often called in vitro recombination. Although several other ways of generating potentially beneficial mutations exist, like mutagenic PCR, DNA shuffling is much quicker and easier. Also, library generated by a mutagenic PCR typically contains 70 to 99% of non-functional variants, whereas most variants formed by DNA shuffling are functional. How it works? Classic DNA shuffling is done as follows. First of all, you need an initial set of mutant genes derived from a single gene. When DNA shuffling is done between closely related genes, instead of a set of mutant genes derived from a single gene, this technique is called DNA family shuffling. DNA family shuffling utilizes the naturally occurring genetic diversity of family genes as a driving force for in vitro evolution. Then we treat the genes with DNase 1. Now what we have is the set of fragments with lengths depending mainly on the amount of enzyme, incubation time, and temperature. Then we need to separate these strands by denaturation in order to get complementary regions available for hybridization. After that, we cool the mixture and let those strands anneal. What we have is a set of hybridized strands like this. Finally, we use PCR without primers to get full double-stranded DNA. Then we can modify 5' and 3' ends using tailed primers in order to incorporate these mutant genes into the plasmids. One difficulty in DNA family shuffling is a strong tendency for this method to reconstruct the initial gene structures. It happens due to preferential annealing of the perfectly complementary strands of DNA fragments derived from the same gene. By using the sense single strand of one gene and the antisense single strand of another gene as starting materials for DNA family shuffling, high recombination efficiency can be expected, as only heteroduplex molecules are formed in the first round of PCR. We can use single-strand DNA family shuffling when conventional DNA shuffling does not yield recombinants at a high frequency due to a low degree of homology between two parental sequences. Although this method generally shuffles two parental sequences at once, more genes can be shuffled if the process is repeated. Genes with beneficial mutations can be shuffled further, both to bring together these independent beneficial mutations in a single gene and to eliminate any deleterious mutations. If you want more detailed video about DNA shuffling techniques, let me know in the comments below.